Rotarians and guests, I have the privilege of introducing our speaker today, Elio Arruzza, a lecturer and researcher in medical imaging at UniSA. Elio is exploring how the Parkinson's changes the appearance of certain brain areas, and by using brain scan markers, a more accurate and earlier diagnosis of Parkinson's may be found. The reason I'm doing this introduction, apart from the fact my brother dobbed me in, is because I was diagnosed with Parkinson 8 years ago, and luckily I've got good control of it. But with 150,000 Australians diagnosed with PD, it's incredibly important research. So welcome, Elio.
Hi everyone, and thanks for having me come along today. And thank you, Di, for the kind introduction. I've never done a presentation after being fed so well. So normally the dinner comes after the presentation as a reward. So yeah, thanks very much for that. So as Di mentioned, my name's Elio. I work at the University of South Australia as a lecturer in medical radiation, or specialise in medical imaging. But I'm also a doctoral candidate in the area of Parkinson's, so a topic that I'm really interested in, and hope to share some research about what we're doing at the university into Parkinson's disease today. So many of you would be aware of Parkinson's disease through, I guess, personally, or through family and friends, or even popular figures in culture or in media, like, for example, Muhammad Ali or Michael J. Fox, these types of people. Parkinson's was first discovered in the early 1800s, so over 200 years ago, and was discovered in London by a doctor named James Parkinson, and he labelled it a shaking palsy. But it wasn't until 50 years later where the same types of patients kept coming through, and they named it in honour of James Parkinson. If we fast forward to today, we know that Parkinson's disease is the second most common neurodegenerative disease after dementia in Australia. About 10 million people, just over, are currently diagnosed with Parkinson's worldwide. About 150,000 people, as Di said, in Australia have been diagnosed with Parkinson's disease. And about 50 people every day in Australia are diagnosed with the condition. Should also note that men are more likely to develop Parkinson's compared to women, so about 1.5 to 2 times more likely. And the general age of onset in most cases is after the age of 60. That's when it's diagnosed. So what does Parkinson's look like? What are the signs and symptoms? There's a wide range of signs and symptoms, and some of you might be aware of the more movement type issues that are associated with the condition. The first one's called bradykinesia, so this essentially just means slowness of movement. So the everyday activities like writing, buttoning a shirt, these types of things take a lot longer. Tremor, so we all kind of have a natural tremor in a normal healthy person, but someone with Parkinson's develops that kind of characteristic involuntary shaking. Rigidity, so this is when the muscles become stiff even if they're not being or wanting to be contracted by the patient, so there's muscle stiffness. Impaired gait, so some people with Parkinson's will develop that kind of characteristic shuffling style of walking that you might see. And postural instability, fancy word, which essentially means difficulty balancing. So those kind of quick unconscious adjustments that we all kind of make when we're walking or trying to stand up, that kind of ability does degrade in someone with Parkinson's, so losing the ability to balance themselves. So those are kind of the more characteristic movement type issues that some of you would be aware of. But if we look at this graph here, it's there's a lot of words, but if I can go through it, it might be better to see there. You can see that Parkinson's has a wide range of symptoms and signs, but a lot of them, and what many people don't know, is that they're non-movement issues. So if we look at this graph, we have time along the horizontal or the x-axis here, and we have the degree of the disability along the y or the vertical axis here. And we can see that there's what's called a prodromal phase of Parkinson's, essentially the early warning signs, and these contain symptoms and signs which are non-movement related. So things like sleep issues, mood issues like depression and anxiety, smell issues, so that's what hyposma is, a loss of sense of smell. And they actually may begin prior to the person having the movement issues. So it eventually gets to the movement issues, so the things that I was talking about before, the slowness of movement and the tremor and those types of things. But what's actually happened is that prior to that time, sometimes many years before, they've had other issues to do with those non-movement. And so as the disease progresses, the non-movement issues get worse, the movement issues also get worse, and then we start seeing some later stage disease signs and symptoms like dementia, issues walking, issues with speech, even hallucinations and that type of thing as well. So basically the point of the slide is, one, that the disease happens over many years, and two is that there's that movement component, but there's also a large range of symptoms which don't involve movement at all. So I guess how does the disease begin and how does it progress? Like we saw in the graph, two of the early signs of the disease which don't involve movement are those which involve smell and digestion. And so what many researchers think is because these are affected, these two kind of functions have one thing in common, and they are exposed to the outside world. So smell is to do with our nose, and the digestion we get through the mouth. So I guess what researchers tend to think is that it's likely due to some form of environmental factor, something like in the food we eat or in the air we breathe, that might play a role in how Parkinson's begins and develops. And so as the disease goes on, we start seeing different parts of the brain involved. And so not only does smell and digestion get affected, but eventually the disease moves on to what we call the midbrain, which is a part of the brainstem, and an area called the substantia nigra. And that's really important because that's the area that's really important for movement and control of movement. And so when the disease reaches that point, we see the issues to do with movement. Eventually, the disease progresses to, holistically more parts of the brain, and we start seeing cognitive, memory and thinking issues, and emotional disturbances as well. And that progression happens over a long period of time. So you might be thinking, well, what might actually cause Parkinson's disease? The answer is we don't really know, but it's thought to involve a variety or a combination of both genetic and environmental factors. So some Parkinson's is, there's a familial basis to it. There might be a family history, but it's probably only 10 to 15% of the Parkinson's cases we see have a family or a genetic background to it. More the environmental factors which we think might be at play, and one which is surprising to a lot of people is certain exposure to pesticides and herbicides. So there's two which a lot of research has been done into. One's called paraquat and one's called rotenone. So paraquat is an agricultural herbicide used on crops, and it's used in the kind of commercial agricultural world. From my understanding at the moment, it is banned in a lot of countries in Europe, in the UK, I think in China as well, but it's still under heavy restriction and legal to use in Australia. So that's one of them, and the other one is called rotenone. So rotenone is an insecticide, and that's used on crops as well, and it can be bought by members of the general public at the local hardware or gardening store through those garden dusts. A lot of the active constituent or the chemical in those garden dusts involve rotenone. And so there's been a lot of research into long-term exposure to those chemicals. And often so much they're quite been proven in a lot of research to be in links with Parkinson's because they're actually used as animal models for the disease. So in a research setting, mice and rats are provided with these chemicals to simulate the effects of Parkinson's so that they can study the disease more. So that's how much that there's a link there. And so the next point, rural living, for those people that might live next to agricultural land or are farmers or live rurally, there's been some studies to show that they're at higher risk of developing Parkinson's disease. We tend not to talk about cause in health or health science. We talk about risk. So is a certain activity or a certain genetic factor is it associated with an increased risk of developing the disease? So it doesn't mean you're definitely going to get it, but it just heightens the risk. Traumatic brain injury, that probably is one where is more commonly known, so repeated concussions and trauma to the brain has been seen to have a link. And the other one is certain illicit drugs, particularly methamphetamine. So methamphetamine affects the part of the brain and the type of brain cells or neurons that are also affected in Parkinson's disease. And just like I was talking about before, in animal models, scientists also do give, a form of methamphetamine to lab animals like mice and rats in order to simulate what Parkinson's looks like in their research setting. So there's also other risk factors, but these are kind of the main ones here. So I guess in terms of research into Parkinson's disease, there's a lot of research going on, and they can be kind of split into 5 different fields. So there's research looking into the underlying disease process of Parkinson's, how we can better protect neurons or brain cells, how we can treat the disease better, identifying predisposition or those that may be a potentially higher risk of developing the disease in the future, and then looking into diagnosis and earlier detection. I'm a radiographer by background, so I work within medical imaging, and my research group's interests and aims are mainly to do with those last two, so identifying predisposition and diagnosis and early detection. And we do that through imaging, so looking at brain scans of a variety of different brain scans. So you might be familiar with some of these words, SPECT, PET, ultrasound and MRI. They're used clinically for those who have been to a radiology department before. And we use some of these different scans in Parkinson's disease research. Particularly, our research group uses the ultrasound and the MRI scan. So ultrasound scan, you'd be familiar with, same ultrasound scan that's used on pregnant women, or if you have an issue with your muscles or something like this. We use the exact same scanner, except instead of putting the ultrasound probe on the person's tummy, we put it just on their temple. Because that part of the skull, there's a bit less bone there, so that the ultrasound scanner can penetrate through. So very safe and non-invasive and painless. We can get really good images there. So we can see a brain there with a cross-section. That's kind of the part of the brain we're looking at. And then the other image there is, oh sorry guys, I feel like I'm leaving you out, is the ultrasound scan being done by my research group supervisor there. So probe just put on the skull there, and we can get a pretty good image of the area we're looking at. So I guess in Parkinson's disease, the part of the brain we're looking at is the substantia nigra, which I mentioned before, and on the ultrasound scan, that kind of lights up bright in people with Parkinson's, and normally isn't bright in someone who doesn't have Parkinson's. However, it sometimes will light up bright in people who don't have Parkinson's, and that is kind of a marker which indicates potentially high risk of developing the disease later in life. So it's a really good tool for potentially seeing those changes. The other one that we use is the MRI scan. Magnetic resonance imaging. You might have had an MRI before, so we also can do an MRI sequence to look at that substantia nigra. And before, I talked about that kind of lights up bright on ultrasound. It does the opposite on the MRI scan. So where we would normally see a kind of marker in someone with Parkinson's disease, we probably don't see that marker anymore. But it can also act as a marker for seeing the risk of developing Parkinson's later in life, that finding. So I guess the aim of our research is to improve understanding of what changes in the brain mean on images, and then how they relate to various body functions. And the body functions that we're looking into are the ones that are particularly affected, the non-movement ones that are particularly affected in Parkinson's on that graph I showed you before. So part of our study is that we're obtaining these images on the ultrasound scanner, and then we're doing a whole bunch of tests and questionnaires about body function and those particular body functions I mentioned. So if you see on the image, you see, for example, someone with headphones on, and so we record their speech to see what their vocal quality and control is like, which is affected in Parkinson's disease. We also do some heart rate and blood pressure testing using ECG leads. And a lot of participants will also get a neurological examination done by a specialist or a neurologist, which is a doctor that specialises in conditions of the brain. And so, yeah, by getting those images and using the body function tests, we can hope to increase an understanding about the risk of developing Parkinson's and potential early disease processes. So I guess the overarching aim is earlier diagnosis and a better look into who might be at risk at developing the disease. I should thank our research group. So we have a wide range of professionals and researchers in our group, from areas like neuroscience, radiology, speech pathology, and a few PhD candidates as well. I've just put my details there in case you wanted to get in touch, email and phone. And there's a QR code which leads us to the current study. So our current study is, like I mentioned, getting those tests done at our UniSA City East campus, where people will come in for about 2 hours, and these are healthy people, so not necessarily those with Parkinson's disease. But what we find in the healthy population, we can kind of transfer the findings and find a little bit about, more about what we might see in the Parkinson's population. So yeah, it's a 2-hour appointment there. Yeah, be very appreciative of your time. If you had any time to come down, I can provide you with some more information about that as well. So I think that brings me to the end. Thank you all for listening today, and very welcome to answer any questions. Thanks. Cheers. Oh, wow. Hi.
Let's see if we ask the same question, Rajiv.
Sorry.
Thank you for your excellent address, and thank you for that excellent introduction, Diana. I just wonder, given that the Parkinson's is incurable, what is the benefit of actually getting knowledge about what's about to come?
Yeah, it's a very good question. It is incurable, like you said, and so what current medication does is kind of masks the disease. So it treats the symptoms, but still the underlying disease processes happen. Although we don't have a cure right now, who's to say in a few more years we do? So what a lot of research goes into is what's called neuroprotective drugs or treatments, and what they do is they preserve what brain cells in the area that you currently have. And so if we can really detect people who might be at further or might be at risk of developing the disease, and we see that early, we can get them onto these neuroprotective drugs, and so it could stop it in its tracks. Just like that graph we saw before, the movement symptoms and the really debilitating symptoms happen after a lot of the early kind of non-movement signs. And so if you can kind of catch someone before a lot of those aggressive movement symptoms come in, and you can not reverse the disease but stop it in its tracks, then that's the benefit of knowing it early. So, yeah, thanks.
Thank you. Thank you. My mum had her DBS surgery in 2014.
Yep.
The effects of it lasted only for 5 or 6 years.
Okay.
So is there any further advancement in DBS which makes it longer lasting now?
Yeah, so
Thank you.
thank you. I haven't done too much research into DBS, but I have had the pleasure of actually being in the room while it's done. I guess for those who might not be aware of DBS, it stands for deep brain stimulation. Essentially what happens is that the neurosurgeon will kind of take off the top of the person's skull whilst the person is still conscious and with their eyes open, and then plant some electrodes and equipment into the brain and send electrical impulses whilst the patient's still awake. And so they can actually do tests with their hands and certain tests with their face and their body whilst they're delivering those impulses. And I'm sorry to hear that it didn't last too long. I'm sure the technology is always getting better. Hearing from people who've had the DBS, they do, and seeing it live, they do go through enormous benefits from it. I'm not sure why it didn't last long, but hopefully there's treatments which can make it last longer. So, yeah, thanks.
Just want to reiterate, thank you very much for an outstanding performance.
Thanks.
It's quite gratifying to hear speakers of your calibre. My question is, okay, so what are the preventive behaviours you can put in place?
Yeah.
You're 60-something, maybe 60-10.
Yeah.
You want to protect yourself. I mean,
Yeah.
the crossword business that people bang on about, keeping mind active. Are they beneficial at all, or is it just something for the newspapers to babble on about?
Yeah, I guess it is a common question. I guess it's like any form of disease associated with ageing. The things that you do to otherwise keep a healthy lifestyle is what you should be doing. So eating well, exercising, regular checkups with the doctor, and seeing them when you have issues. There's not a certain antidote for Parkinson's particularly. There has been some research, and it's to do with these risk factors as well. That like drinking coffee might reduce the risk of developing Parkinson's. So for those coffee drinkers out there, but they just might be some single studies here and there. The main thing is just to look after your health like you would in other, ordinarily, and that reduces your risk of all different other chronic conditions as well. So, yeah. Yeah.
Sorry, just a very quick one. And I agree with the other comments. Excellent address. I think there'll be no one in this room who hasn't experienced through a friend or family or associate Parkinson's. So I think we're all well aware. I'm certainly well aware of the issues in agriculture because we have agricultural properties. I know that in southwestern Victoria, with the potato growing, there's quite a focus on Parkinson's and the use of chemicals. I was going to ask, I've got a son who's very strong on the idea of the benefits of the nicotine. Now, that doesn't mean taking up smoking, but nicotine on the brain apparently is very good. I wonder if you want to comment on that.
Yeah, look, I wasn't, just like I was reluctant to mention the coffee thing there. In the same way I was reluctant to mention the smoking. I think there's been a handful of studies to show that smoking might reduce the risk of, or the nicotine might reduce the risk of developing Parkinson's. I probably wouldn't take on that advice just because, although it might reduce that risk, it probably...
You don't have to smoke to get nicotine though.
Oh, I see. Yeah, that's a good point. I would have to look a bit further into that. But yeah, it's an interesting area of research if we can determine that. Yeah.
Okay. Another question?
Yeah, thank you. Thank you for a great presentation. Firstly, Parkinson's, one's difficult to diagnose and often misdiagnosed. And your research was on the two things of ultrasound and MRI, and you stated that an ultrasound, it shows up bright, and in an MRI, you can't see the substance nigra. So I just wondered, what's the physiological, biochemical, radio thing? Why do those two things occur? And is this your aim to get an early diagnosis?
Yeah, so I guess the reason why it happens is that that area of the brain has a lot of iron in it. And so the reason why it kind of lights up on the scan is due to the content of iron. We don't know too much about what's happening, whether it's increasing or decreasing, but we know that the reason why it's lighting up is to do with iron. And so the ultrasound essentially, like a sonar, it reflects what metals in the area, and so the more metal or the more iron, it gives a higher signal. So yeah, just the second part of the question, we're doing a lot of research now, particularly the other members of the group, into hopefully using the MRI particularly as what we call a biomarker. So if we see that in the clinical setting and we see it early, then we can diagnose it a lot earlier, because it is quite specific to Parkinson's. So we see it in Parkinson's, but we don't necessarily see it in other brain conditions, which hopefully leads to a decrease in the misdiagnosis rate. So, yeah.
So why is it dull in the MRI?
Oh, that's just to do with the properties of the scan. On one it lights up, on one it disappears. It's just to do with the contrast of the scan. So sorry if I didn't make that clear, but essentially the finding in the ultrasound is the brightness. In the MRI, it's still different to the healthy population, but it's the disappearance of the area on the scan. So.
We're running out of time, so I'll be very quick.
Oh, sure.
And my question leads from the last question. I'm a PhD doctor, so I have no idea about medical, but my father's love of hospital has led me to read quite a lot about this.
Yeah.
This has to do with ultrasound machine.
Yeah.
So what I've read, and this is what in my mind was, that ultrasound may not be specific enough to differentiate between Parkinson and neurodegenerative disorders. So how do we ensure that we pinpoint that the diagnosis is correct? Second question about ultrasound, that it may not provide details, images for the brain structures that affect the Parkinson, which is substantia nigra.
Yeah.
So how do we ensure that when we are diagnosing it, we take those two into consideration?
Yeah. So I guess with ultrasound, it's good for many parts of the body. The irony is it's quite terrible for the brain, right? But it's really good for this particular part of the brain, the substantia nigra. So it's not really used in a clinical sense for the head, but in our research we can use it, and we use it just to look at that substantia nigra, not any other parts of the brain. The first part of the question was, can it differentiate between other neurodegenerative disorders?
Yeah, and Parkinson ultrasound.
Yeah. Well, the key thing about Parkinson's is that it really affects the substantia nigra, which other neurodegenerative disorders won't affect, so or not as much. So the fact that we're looking at that bit only means we can use it quite specifically for Parkinson's disease. But in terms of the entire brain, you're right, the ultrasound's not that great. It's really just good for that particular area. And that's where other imaging modalities come in, like MRI, which gives a holistic view of the brain a lot better than what an ultrasound would.
Thanks for clearing that up.
Yeah.
From past many months, every time I do research on ultrasound, substantia nigra comes up. So I said, why?
Yes.
Why are we still using that machine that clears up? So thanks.
Yep, that's why. No worries. Oh, hello.
I think you might have answered my question. I was wondering what the linkages and the similarities are between early Parkinson's and Alzheimer's.
Yeah, I guess they're two different degenerative diseases. My study's mainly been in Parkinson's disease, so I can't really talk too much about Alzheimer's particularly. But like we've mentioned before, there is a lot of crossover between certain neurodegenerative diseases. Parkinson's is fairly relevant because it's got that movement characteristic, and so a neurologist should be able to pick up Parkinson's compared to something like Alzheimer's, which normally happens at a later stage. So, yeah, sorry I can't really answer that really well, but hopefully that little bit helped. Yeah.
This is a question a bit like the nicotine question.
Yeah.
In relation to LSD and psychedelic drugs, has there been much progress done in that area? Because I once watched a program where this person was given LSD and, it's Parkinson's, and then within like a very short time was doing cartwheels, and then
there you go.
not long after it was back to square one.
Yeah, right. Was that on SBS? Maybe something like that. Generally the connections between the drug use or the certain research that we've done is that it actually can assist in causing Parkinson's rather than treating it. Things like methamphetamine, for example. I'm not sure, I haven't seen that study. It's an interesting one to look at. So I probably can't comment too much on LSD. Oh, okay. Right, I see. Yep. Dopamine's the main one in Parkinson's disease. Serotonin is slightly affected as well. So it might help with the symptoms of Parkinson's disease, of the body functions that rely on that chemical, serotonin. But in terms of the dopamine itself, it certainly wouldn't be able to reverse the disease or cure it. Yeah.
Well, we grant you a PhD now.
Thank you. Well, tell the uni that. Thanks very much. Thanks, Don.
Thank you very much. And I think there's something small that looks like it's just for you.
Oh, thank you. Oh, thanks so much. I'll grab that. No? Thanks so much.
I can't reach it.
Well, that's all right. Cheers.
Yeah, thank you very much.
No worries.